So, class, I welcome you back into another important lecture. And for today's class, we will have a look on toward the ribosomes and on the endoplasmic reticulums that we have. And in the endoplasmic reticulums, further, we would see the structure and the function of the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Now you see that here we have got these spherical structures, and these spherical structures they are entitled as the ribosomes. As of the previous class where I said that nucleus is the largest organelle that we have got in the cells. So likewise, the ribosomes that we have got, so these would be the smallest organelle, and an important other feature of these ribosomes is that unlike the nucleus and the mitochondria and the chloroplast, they are not bound by any of the membrane. So they are non-membrane bound organelles. And if we were to talk about it, what would be making up these ribosomes? So you see that to the nucleolus of the nucleus, when the ribosomal RNA and when the protein they would be assembled together, they would be linked together, then we would make these ribosomes out of it. These ribosomes would go out of the nucleus and then they would attach themselves to the membrane of the rough endoplasmic reticulum, or they can disperse themselves freely in the cytoplasm. Now, further, what we have to see is that wherever the ribosome would be discussed, so these ribosomes they are further made up of two subunits. Okay, like there would be total of two subunits that we have in them. Among them, they would be coming up of a smaller ribosomal subunit, and then there would be a large ribosomal subunit. Like if I were to show you the diagram, so you see here, like for the ribosome, I have this large ribosomal subunit. I have this small ribosomal subunit. And in between them, there would be the coming up of a groove that we'll see in the context of the protein synthesis chapter. Now, overall, if we try to be looking on toward the function that these ribosomes they perform, so they would be making the proteins. They would be assembling the amino acids. And how the amino acids they are assembled? Because genetic information would be copied on the messenger RNA, and that messenger RNA, when it comes to the ribosomes, on the ribosomes amino acids they would be assembled together, they would be linked together by the peptide bond, and by that we would make the protein out of it. Overall, we try to be splitting these ribosomes into two distinct types. Like we have got some large ribosomes and they are the 80s ribosomes, and then we have the small ribosomes and they are the 70s ribosomes. So there appear some differences among them that we need to look on. These 80s ribosome by size they would be 25 nanometer in diameter, and comparative to the 70s ribosomes they are larger. And where they would be located, they would be found within the cytoplasm, and they would be available on the membrane of the rough endoplasmic reticulum. Now, this word S that we have got, this stands for Svedberg unit. The Svedberg unit simply means, like if I were to isolate the ribosomes and I would take them in a test tube, and if this test tube was spun at a very high speed, so more heavier or the larger ribosomes that we have, so they would settle faster to the bottom of the test tube. And for that, the eukaryotic ribosomes that we have got attached to the rough endoplasmic reticulum and that we have got in the cytoplasm, they are the heavier kind of the ribosome, because when we would spin them up at a very high speed in a centrifuge, so they would deposit faster as compared to these lighter 70s ribosomes. And you see that these 70s ribosomes comparative to the large ribosome, their size again is small, and this is 18 nanometer. And where they are found, all of the bacteria that we have, okay, like the kingdom prokaryota that we have, which includes all the bacteria. So these ribosomes, they would be found there. But there is some unique thing that the mitochondria that we have in the animal and the plant cells and the chloroplast that we specifically has got in the plant cells, so they have their own ribosomes, and these ribosomes they are not 80s ribosome but smaller 70s ribosomes. So the eukaryotes also have the smaller 70s ribosomes, but only in their mitochondria and the chloroplast. But all of the bacteria they would have these 70s ribosomes in them. Now let us just try to see their color diagrams and how they appear under the microscope. So you see here that I have this nucleus with me. And in close association with the nucleus, now you see we have got these flattened compartments. These are the rough endoplasmic reticulum. So on the membrane of the rough endoplasmic reticulum, you see that we find ribosomes here. Okay, and then there would be some other flattened compartments coming. They are the smooth endoplasmic reticulum. We will see them in a while. And when we try to look these ribosomes under the microscope, so you see that these are the membranes that we have got of the rough endoplasmic reticulum, and these small dots that you see, they are actually the ribosomes that we have on the RER. And the same way, these small spherical units, they also appear in the cytoplasm. So this is how these ribosomes, they would appear under the microscope in the photomicrograph. Now, moving onward, we have to be discussing about the endoplasmic reticulum as of the before diagram. So you see that when the scientists, they started to look the cells at very high magnifications, so they basically were amazed that within the cells we have got these flattened compartments, and these flattened compartments technically they are referred to as cisternae. Okay, and we have got two type of the endoplasmic reticulum. Here you see that this endoplasmic reticulum that lies in very close association with the outer envelope of the nucleus. So this is a rough endoplasmic reticulum. The rough endoplasmic reticulum is so called because we have got ribosomes attached to them. And here again, we have got these flattened compartments, cisternae, and they have a smoother appearance. You see, like I don't see any granular appearance of them because these smooth endoplasmic reticulum that we have, they don't have any ribosomes on them. So that is why they are entitled as the smooth endoplasmic reticulum. And here we have the granular appearance of these membrane, and this is due to the ribosome. So this is rough endoplasmic reticulum. So now you see that these rough endoplasmic reticulum, so they are extensive and they are a connected system of the membranes. And these flattened membranes that we have got, so we call them as cisternae, and they would lie in continuous pattern with the nuclear envelope of the nucleus, and they would run through the cytoplasm. And on these rough endoplasmic reticulum, there would be ATS ribosomes attached to them. So overall, if we just try to justify the function of the rough endoplasmic reticulum, now since they have got ribosomes attached to them, so they would be assembling the amino acid, and then we would make the proteins out of it. But the thing to remember at this point is that any of the protein that is formed on the ribosomes of the rough endoplasmic reticulum, they are the rough protein. Why we call them as a rough protein? Because their function is yet not decided, that whether they are some enzyme, whether this is a keratin protein, whether that is a collagen protein. So this has to be decided later. So the other role which these rough endoplasmic reticulum they perform is that after the rough protein has been made, now from these rough endoplasmic reticulum, some vesicle would pinch off, and they are the RER vesicles. And the RER vesicles, they would take the rough protein toward the Golgi body. And over there, we would try to modify the protein, like by causing the protein to fold up, or we can also attach the short carbohydrate chain to these proteins, and that would be in term of protein modification. But that protein modification does not happen on the rough endoplasmic reticulum, but rather than it happens in the Golgi body. But in term of the protein modification, we can say that these RER, they package up the rough protein in these RER vesicles, and then they are carried to the Golgi body for the modification. Further moving on, we have got the smooth endoplasmic reticulum, and these smooth endoplasmic reticulum, they are so called because I have shown you in the diagram that they do not have any ribosomes on them. Then the idea is that what functions these smooth endoplasmic reticulum they would be performing. So the simple thing is that these SCR, they are the site of the lipid and the steroid synthesis. Okay, by the steroid synthesis, what we mean is that we make some steroid hormones. Okay, and in the steroid hormones, we have got two of the major hormones to be discussing about. One is a testosterone hormone, and the other is a estrogen hormone. And these hormones, they are formed within the smooth endoplasmic reticulum. Why these hormones they are important? Because they determine or they develop the male and the female secondary characteristics. Like when we reach the age of puberty, then there happens to be the growth of the hair under the armpits, on the face, and then our body, it gets broader. So these are all the male and the female secondary characteristics. So they are under the impact of these hormones, and where they are made in the smooth endoplasmic reticulum. Now moving onward, we will see in your E2 biology that in the muscles we have a specialized kind of these smooth endoplasmic reticulums, and they are termed as sarcoplasmic reticulums. And what these sarcoplasmic reticulum they do is that they keep in themselves the store of the calcium ions, and these calcium ions are important for the contraction of our muscles that we will see later in your E2 biology. But for the moment, you just have to see as that they are the site of the lipid and the steroid synthesis. We make the testosterone and estrogen hormone here. They would act as a source of the calcium ion in the muscle fibers. And then another important feature is that the smooth endoplasmic reticulum I am talking about at the moment. So they also have got a role in drug intoxication. Okay, because whatsoever the drugs that we take up in the daily life, so they produce some toxic metabolites. So they are intoxicated within these smooth endoplasmic reticulum I'm talking about. Now moving onward, here we have to be talking about another important organelle coming. And this important organelle is what we call this as the Golgi body. Overall, the role of the Golgi body is that it would be modifying the proteins. And in terms of the protein modification, you need to have grip on toward the keywords. One of the keyword that I would be using later in terms of the Golgi body, this is glycoproteins. If I produce a protein on the ribosomes, I pack them in the RER vesicle, and when I'm bringing them to the Golgi body, and if I attach the short carbohydrate chain to these proteins, so it would become glycoprotein. Likewise, if I have the protein and if I don't attach the glyco part, but rather than if I attach the lipid part to the proteins that we are bringing from the RER, so it would make glycolipids. Okay. Now, I were to show you this situation. You see, here I have got this nucleus. The nucleus would be making the messenger RNA. Messenger RNA is coming toward the ribosomes, and here we have made the protein. This is a rough protein. The rough protein is being packaged up in the RER vesicle. And now you see that that part of the Golgi body where the RER vesicle is fusing with. So this would make up the cis face of the Golgi bodies. And that part of the Golgi body from where after we have modified the protein and we pinch the Golgi vesicle out having a final protein into it, so that would be the transphase that we have got for the Golgi body. In terms of the Golgi body, we use another word which is Golgi apparatus. And for that, if I have got these Golgi bodies and if I include the Golgi vesicles along with it, so ultimately that would be making the Golgi apparatus. Now, another color diagram is here. You see, this is again the nucleus. Here we have made the messenger RNA. Messenger RNA would be leaving the nuclear, will be leaving the nucleus to the nuclear pore. And here you see we have RER coming. Okay, rough endoplasmic reticulum coming. And here you see this is RER vesicle. This is carrying the rough protein to the cis space of the Golgi body. And when the protein would be modified here, so the Golgi vesicle would pinch off having the final protein into it. So this is being pinched off from the transphase. And now these Golgi vesicles, by having the final protein into them, so they would be fusing with the cell membrane, and now the protein would be released out of the cells. Okay, so now we intend to look at what roles or functions these Golgi bodies they are performing. So the situation is that any of the protein that I make in my body, no matter if I'm making some enzyme, I'm making some keratin protein, collagen protein, so all those protein in their rough form, they would have the methionine amino acid. And when they would come to the Golgi body, so the Golgi body would remove the methionine from any of the protein which is coming from the rough endoplasmic reticulum. The other word was glycoprotein, as I have said earlier. So if the protein comes in the Golgi body, and if I attach some short carbohydrate chains to it, so it would make the glycoproteins here. And these glycoproteins, you see, we have got multiple examples of them, like the mucus that is being released in our airways, a sticky kind of a material. So this is glycoprotein, and where this is being formed in the Golgi bodies. Afterward, you see that we will come later to be discussing some structures within the animal cells, and they are the lysosomes. And the role of the lysosome is that they have some enzymes in them, and they are the hydrolytic enzymes. Why we call them as a hydrolytic enzymes? Because they break down the complex ones into the simple ones with the use of water. So if I just simply cause some enzymes to be packaged into the Golgi vesicle, and I don't allow the Golgi vesicle to be splitting the enzymes out of the cell, rather than if I try to retain this Golgi vesicle within the cell, So now this would become a lysosome, and this lysosome has got multiple functions in terms of removing the damaged organelles and other stuff that we will see later. Now, the other situation is that we also would like to be making the lipoproteins. Okay, lipoproteins mean that where I am supposed to be linking the lipid component to the protein component. So that would make the lipoprotein. So the situation is that firstly, we would be making the proteins, and when those proteins they would be coming toward the Golgi body. So over there, we would be attaching the short, you could say, lipid chains to the protein, and as a result of that, we would make lipoproteins out of it. So the lipid initially they would be metabolized in the smooth endoplasmic reticulum, and then they would be brought to the Golgi bodies, and over there, when we would attach the lipid to the protein part, so we have made the lipoprotein out of it. The other situation is that in case of the plant, you see that we are required to be making the cell wall. Okay, and those cell wall making it would require some enzymes. So those enzymes again they would be modified within the Golgi body, and then they would later contribute a role in the cell wall synthesis. Other than that, in our respiratory system and in our gut, there is production of the sticky material which is mucus, and this is glycoprotein in nature. So once we bring the protein to the Golgi bodies, and when we attach the short carbohydrate chains with it, so it would make a sticky substance, and that would be the mucin. And afterward, that mucin, when this is released out of the cell, so it would appear in the form of a mucus, and that mucus is a sticky substance that basically traps some harmful particles that we inhale in the air. Now, if I were to simply summarize this, that how come the production or the secretion of the protein would be taking place? So initially, there would be the ribosomes coming. The ribosomes, they would be attached to the RER vesicles. Now here the transport vesicles would take the rough protein to the Golgi body. The Golgi body would modify the protein and it would be packed in the excretory vesicles or the Golgi vesicles. And these Golgi vesicles, they would move toward the cell surface membrane and ultimately they would be released out. How you are supposed to be remembering these steps? You would say that initially the protein synthesis happen at the ribosomes. Okay, like it's happening here at the ribosomes. And then transport vesicles, like RER vesicles, if we just call them. So they would pinch off and then they would fuse with the cis phase of the Golgi body. Okay, like here is a cis phase of the Golgi body. And then here the protein would be modified, and then ultimately the Golgi vesicle would separate itself from the Golgi body, and now this has become the Golgi vesicle. The Golgi vesicle would go further and it would fuse with the cell surface membrane, and now the protein would be released out by exocytosis. So this is what you have to remember in context of the ribosomes, RER, SER, Golgi apparatus. So see you in the next class.